Hi everyone, my name is Akshay Agarwal and I will be presenting our work on predicting epitopes for SARS-CoV-2. This work was funded and done at IBM Research and would not have been possible without the support of all the co-authors. So first of all, to start with some background, epitopes are short amino acid sequences that define the signature of an antigen to which an antibody binds. In this work, what we are doing is that we take known epitopes from SARS-CoV-2 and other coronaviridae and use that to identify additional antigen regions in SARS-CoV-2 genomes. We mainly analyze and discuss spike glycoprotein and nucleoprotein since those are proteins of interest. We also use NetMHC 4.0 to predict MHC classes for T-cell epitopes. Lastly, we analyze the presence and occurrence of epitopes across SARS-CoV-2 genomes and cluster them by sequence and position. So for our data, we relied on IEDB for downloading linear B-cell and T-cell epitope data. This was downloaded first in July 2020 and then again in April 2021. For our genome and protein data, we relied on IBM's functional genomics platform, which is maintained by my team, and we had 62,000 genome sequences and approximately 29,000 unique protein sequences. So without going into too much detail about the data, I just want to point out that at the time of download in July 2020, we had a significant number of sequences, epitope sequences that came from SARS-CoV-2 itself for both T cell and B cell. And most of the epitopes actually belong to spike glycoprotein and nucleoprotein, again, both for T cell and B cell. So once we had downloaded the data, we decided to predict epitopes. So the first one was we decided to identify conserved epitope sequences. And second, we identified candidate epitope sequences by blasting known epitopes against protein sequences. For BLAST, we only kept results that had an e-value of less than 0.01. So we observe here that spike and nucleoprotein sequences have a high number of conserved and candidate epitopes for both B and T cell. We also noticed that complete sequence homology is found only with respect to SARS-CoV-2 and SARS epitopes. That is, no epitopes from other coronaviridae were found to be present in SARS-CoV-2 genomes when performing an exact sequence match. Once we had this set of conserved and candidate epitopes, we passed it through NetMHC 4.0 with a set of 12 most commonly used alleles to identify MHC classes for T-cell epitopes. So even though we performed result verification very late in our analysis, I would like to actually start with it. So here we downloaded data again from IEDB in April 2021 and blasted our set of candidate epitopes against this set of IEDB epitopes. And we found that the average percentage identity across all sets is greater than 90%. This gave us a lot of confidence in our findings. So one of the first things we had done after identifying conserved and candidate sequences is that for each class, we performed sequence-based clustering. This analysis was to help visualize or analyze which epitopes are clustering together by sequence and how the presence of these clusters vary across genomes. So in this figure, in the bottom of the figure, we show hierarchical clustering of nucleoprotein T cell epitopes. These includes both conserved and candidate epitopes. In red are the SARS epitopes, green indicates the SARS-CoV-2 epitopes, and blue indicates the predicted epitopes. In the top part, we show presence of clusters across our entire genome set. It's a log-log stacked bar plot, where red indicates presence across animal genomes and green indicates human genomes. Here I would like to point out two things. So in the bottom of the figures for each cluster, if we look at the candidate sequences indicated by blue, it's possible to quantify the expected rate of change of an epitope. This can be done more accurately if the snapshot is taken at different points in time. Also at the top half of the figure, we see that certain clusters have significantly more presence than others and thus might be more useful. Next, we move on to construct the immunodominance plot and the mutation density plots. So for this, again, we constructed this plot for all classes, that is spike, glycoprotein T cell and B cell, and nucleoprotein T cell and B cell. For spike, we looked at approximately 4,500 sequences and mapped the normalized immunodominance, which is indicated in figure A, and normalized mutation density, which is indicated in figure B. Now, from figure A, we can observe that immunodominance is fairly uniform except for two upticks in the S2 subunits of spike. We also see very low epitope presence in the middle. In figure B, we observe that mutation density is a bit high in S1 subunit. Results like this are useful to see which regions are most stable as well as have high immunodominance and could be utilized in downstream analysis such as selecting candidates for epitope-based vaccine. So based on the epitope presence across 69,000 genomes, we report the top 10 epitopes for spike B cells, spike T cell, nucleoprotein B cell, and nucleoprotein T cell epitopes. We also report the number of genomes they were found in, other epitopes that they clustered with based on sequence, the start position, and the parent epitopes in case this epitope is a candidate epitope. For T cell epitopes, you'll observe that we mark some of the epitopes with an asterisk. So this asterisk indicates that it's an MHC class 1 epitope. For our top 10 epitope list, we did not rely just on net MHC to identify the MHC class, but we actually manually checked it against IDB. One interesting result that we found was that net MHC predictions listed fewer epitopes as class 1 epitopes as compared to our manual checking from IEDB. So we also cluster epitopes by position. In figure A, we indicate the epitope position across the protein, and we also mark the top 10 epitopes. In figure B, we list the top 10 epitopes, the 3D structure of the protein. Again, there are two subfigures in panel B and C, one indicating the pre-fusion state and the second one indicating the post-fusion state. So here we look at the number of epitopes that are present across genomes over time, and we see that there's very little change as is indicated by the very small negative slope that is there. Since above data is only till Jan 2021, we downloaded reference protein sequences for different variants and verified that the same trend continues. We also checked whether the epitopes are present in protein created by mRNA vaccine sequences, and we found all of them to be present since these mRNA sequences have high homology with reference by glycoprotein sequence. So in terms of results, so from the number of exact overlapping epitopes or exactly matching epitopes, we can say that there is a high homology between SARS-CoV-2 and SARS-1. We observe that a large number of stay epitopes are present, which is a strong signal for vaccine development and other analysis. We also report top 10 epitopes on spike glycoprotein and nucleoprotein. We discuss a method to calculate probability of evolution of epitopes, and we report that there is high epitope conservation on mRNA sequences and on genomes across time. Lastly, if you would like to read the preprint of our work, please go to this link or scan the QR code. Thank you.